you all and tell you a little bit about what I do and what I've done on Mosaic, and happy to answer questions on any and all topics related to that. So my research centers primarily around climate and carbon, but specifically the impacts of microbes on those two environmental factors. And I work mostly in the Central Arctic Ocean. However, the lab that I'm in at Scripps, which there it goes. Okay, the lab that I'm in at Scripps is the Bowman Lab. I work with Professor Jeff Bowman, who is a microbial ecologist, and our lab actually does work all over the globe, which is kind of cool. The thing that ties all of our research together is that we use a lot of the same methods, but we have work going on from the Arctic to the Antarctic to coastal California to Ecuador and Utah, everywhere pretty much. And all that work is centered here at Scripps. There's the website for the Bowman Lab if you want to learn more. And I joined the Bowman Lab in 2018 now. I've been here for quite a while. I'm actually wrapping up my PhD hopefully by the end of this year. So I'm nearing the end of that. But before I started work in the Arctic, I actually have a background in Antarctic environmental science. So I went to school at Boston University for Earth Environmental Sciences for my undergrad, where I did work on paleoclimate in Antarctic lakes. So I have a bit of an interdisciplinary background going from strict paleoclimate and geochemistry over to microbes, which is the thesis that I'm working on now, looking at microbes in the polar regions as well as biogeochemical cycling. And anytime I mention the word microbes, because there's lots of different definitions, but what I'm referring to is basically the consortium of microorganisms that can be found both in the water column and within sea ice. And I'm just referring to anything smaller than 60 microns, which for reference is about the width of a human hair. So teeny tiny, need a microscope to see them, but they're actually doing a lot of work. And what's interesting about them is that they're not only highly abundant on our planet, they make up actually the bulk of biomass on our planet. So if you were to take a drop of seawater from the coastal ocean, there's almost 1 million bacterial cells just in that one single drop. So they're everywhere. They're doing lots of important things, but they're also really diverse. So we don't know most of the diversity associated with microbes, particularly the bacteria and archaea. So here's a recent image of the taxonomic phylogenetic tree. And so there's these different branches. The eukaryotes branch, where we are, is super tiny in comparison to the diversity of life present within the prokaryotes, so our bacteria and our archaea. And they're not only diverse taxonomically based on who they are, but also in what they're doing. So they play a huge role in biogeochemical cycling on our planet. For example, they produce about 50% of the oxygen that we breathe. So every other breath is coming from the ocean, from our microbes there. And they, microbes on our planet, both in the ocean and on land, do about 90% of the nitrogen fixation. So important for things like fertilizer and crops and everything like that. So they're small, but they're mighty. They're really doing a lot of the elemental work on our planet. And because they're so small and they are in a really unique position to not only respond to their environment, so they're responding to the environment around them, but they're also exerting influence on their environment through things like biogeochemical cycling. And this unique position makes them a really sensitive indicator of environmental change and ecological function or ecosystem services. So really what our planet is doing. And so that's one of the reasons why I'm so interested in what they're doing in the Arctic. So as the Arctic changes, they could potentially be a really sensitive indicator for that change. But the ones that I'm primarily interested in, there's a lot of, as I mentioned, they're really diverse. They're doing lots of different things. But the microbes that I'm the most interested in are those relating to the carbon cycle. So I'm really interested in our phytoplankton, which are our more plant-like microbes, which are doing photosynthesis and fixing all of that atmospheric carbon into their cells, and providing a source of food for the rest of the Arctic food web as well as our bacteria and archaea, so our prokaryotes. However, while they have the ability to cycle lots of different of the important elements for life, I'm primarily just interested in the ones that are more animal-like, more like us. So they're actually respiring instead of photosynthesizing and breathing out oxygen, they're breathing it in just like we do. So looking at this balance, I'm really interested in how much, essentially who and how much are they breathing? That's the key questions that I'm interested in answering in my research. So who's there? How much are they photosynthesizing? How much are they breathing? And what does that do for carbon dioxide, both in our atmosphere and in the ocean? And I'm interested in carbon because of climate change. So I know you've been talking about this both yesterday and today, but the Arctic is changing, and a lot of that has to do with the anthropogenic CO2, which is entering our atmosphere. So as we burn fossil fuels, we increase the amount of CO2 in our atmosphere. This is important to note because it is a greenhouse gas. So we have our greenhouse warming effect, where it traps in energy from the sun and helps to warm our planet. And I think I saw this in one of the lesson plans or for your homework for your after activities, but not all warming is equal. And in the Arctic, we do have something called Arctic amplification, where the changes in warming are twice as fast as anywhere else on our planet. And so I bet if you check out some of those activities, then you'll learn more about why. But what this means is that there's a huge change in the amount of sea ice that's in the Arctic. And for an ecosystem which has been built on having sea ice present year round, reducing that sea ice extent can have huge effects for a lot of the physical processes that are happening there, weather, climate, things like that, but also our ecosystems. And so when we talk about the Arctic, we're really talking about an ecosystem in flux. It's changing really rapidly. And this happens from the scale of people and polar bears and seals and everything that's really cute and fuzzy, all the way down to our microbes. And so, as I mentioned before, if we're interested in these microbes as a potential, indicator for future change, we need to understand what's going on with them throughout this change. And there's been growing consensus within the scientific community that it's actually really critical that we constrain some of our uncertainties in microbial responses to warming, because they are then, cycling those elements, cycling that CO2. And it's clear that microbes in the Arctic could play pivotal roles in feedbacks that actually magnify the impacts of Arctic change. And this is from some things as simple as if you have green algae growing on ice, it's going to be darker. And so it's going to absorb more sunlight. It's going to warm up faster. It's going to melt faster. So a very simple effect, all the way to, how much are they fixing nitrogen or cycling carbon through this ecosystem, and how does that then impact the greenhouse effect and beyond. So there's a lot of recognition of the importance of these organisms, which is why we study them, and which is why we studied them on Mosaic. I see a message in the chat. I'm just gonna check. Oh, yep. So on Mosaic, it was an international Arctic expedition. And what that means is that there was a lot of different countries involved. And so the workhorse of this expedition was the research vessel Polar Stern, which is a German research vessel built by the Alfred Wegener Institute. However, there were almost 20 countries that were involved in sending scientists and researchers for this expedition. And what's really important about this expedition is, not only did we study microbes and biogeochemistry, but we also studied physical processes and atmospheric chemistry and all of these different measurements from different researchers of different disciplines, which can hopefully come together by taking measurements in the same time at the same place in order to put together different processes. So I guess in science, we really like to draw arrows. That's one of our favorite things to do in schematics. But the reason is because we really don't understand how a lot of these different aspects connect. So the goal of Mosaic was to put numbers onto those arrows and try to understand how the aerosols are impacting clouds, and how the cloud impacts sunlight, and how the sunlight impacts the phytoplankton growth, and how the phytoplankton growth impacts the food web. And bacteria, and then they're cycling, and then that cycling then goes back into the atmosphere and everything in between. And so we took measurements, I think, from over 4,000 meters deep in the ocean to 20,000 meters up in the atmosphere and everything in between to try to constrain processes across this ocean, ice, and atmosphere continuum. And the cool thing about Mosaic is that we actually did this for a full year. So this expedition took place from 2019 to 2020, including polar night, which is very rare. We just don't have a lot of data from the Arctic in winter because it's hard to get to, especially the central Arctic, which was our target here. And so what they did was they sailed the polar shear north, froze it into the ice, cut the engines, built an observatory to study all these things, and then drifted with the natural ice drift for the rest of that year, which was, you know, a very intense operation, complicated a little bit by COVID, but we did it. We had people coming on and coming off. It was separated into these different legs of the expedition. I myself was on legs 4 and 5, so this blue and purple part. And I was lucky enough to be there during Arctic summer, so it wasn't quite as cold, but it was a really unique experience in that it was such an extended journey to try and really capture that seasonal transition as well. And yeah, I guess the more stats for fun, there's over 70 tons of equipment that was deployed, over 1,000 ice cores that were collected. And while we were there on our little ice observatory, which this is the schematic, we saw over 20 polar bears and consumed over 2,000 chocolate bars over the course of this expedition, which if you're working in the Arctic, which some of you might have experience with, chocolate is, I think, the most critical fuel source for actually conducting that research. And so, yeah, this is what our ice observatory looked like in schematic. This is closer to what it looks like in real life. So if we have our ship, then we have our different camps. So we have our meteorological measurements, our remote sensing measurements, ice coring, ship-based work in the water column. Some people's job was literally just to walk along the ice and take measurements every day. So they go around the same tracks and see how the ice changed, if it was melting, if there were ponding, if there was ridging. Ice is really dynamic, especially in the central Arctic where there's not really as much of a land influence. So it moves around a lot to the detriment of some equipment. But it was a really, I think, interesting way to try and capture all of that in a targeted framework. And so we were really on the ice quite a bit. So yeah, here's this ponding, trying to move instruments out of melting through. Here's what it looks like when you take a section of ice to core it, collecting water from both the ship and cracks in the ice. So if we're standing on about, a meter of ice here, then goes down to 4,000 meters of ocean underneath. So that's why we're all in these red suits. They're also not only to keep us warm, but they're also flotation devices. And this gave us a really unique chance to collect samples, which it's actually quite difficult to collect from normal ship-based work. So one of the perks of sampling on Mosaic is that we were able to get a lot of ice samples. And so we were able to take newly forming ice, really old ice, sample in pond environments, which are very ephemeral. They can disappear or melt through pretty quickly. So we were able to watch those transitions. And then we had dedicated coring sites, which were sampled for the entirety of the year to see how the ice changed from spring to summer to fall. And the measurements that I'm working the most closely with are from the water column. So one of the ways that we measure the deep ocean is through this instrument called a CTD rosette, and CTD stands for conductivity, temperature, and depth. So you're measuring salinity, you're measuring temperature, you're measuring where you are in the water column, and then you can add other sensors as well. But the key thing is that all these sensors are surrounded by these big bottles, which are essentially columns. And so if you put it in the water, you put them in the water open, and as it goes down, the water can go through it. And then when you get to the depth that you want to measure, you close the bottle caps, and then you're able to take that parcel of water back to the surface. So you're able to measure water that's 1,000 meters deep without having to actually go down and collect it. And this requires a lot of maintenance in the Arctic. This is the base of most oceanography. However, when you're dealing with an Arctic environment, you have to first access the ocean. So there was actually quite a bit of effort into creating an ice hole to put the CTD down and then maintaining it throughout the season. And in the winter, it was covered by a little tent to keep everything from freezing. And so once the CTD gets back on shore, all of the scientists go through and take all the water that they want. And actually, there's pretty much an ordered measurements. You do gases, then everything else, because the gases are the most sensitive. But this was an interdisciplinary effort. So in this video that I'm showing, you can see members of Team Ocean, Team Biogeochemistry, and Team Ecosystem all collecting water from the same rosette, which is why it takes so long. So this is a time lapse, but it usually takes about 4 hours to sample completely, if you're maximizing how much water you get. So this is what I was mostly working with. So I was interested in, again, that who and how much in the water column. And so this is what it looks like for my analyses. So after I collect my water, I put it in a little cooler and I take it with me to the lab. And this is the lab space that we had on board. And so you can see all these different wires. It's because we're a chemistry analytical lab. And I was also doing some microbial work in my little corner over here. And so here I'm going in between filtering the water. So you can see the water going down, and we're filtering it through this really fine mesh, which is collecting all of the microbial cells. So we're essentially accumulating the biomass that's in the water into one condensed location. And from there, we use chemistry to extract the DNA and see who's there. So similar to Ancestry or 23andMe or any of those, I'm doing that same thing, but with microbes. So I'm looking at the DNA and tracking who's there. And then while that's happening, my lab mate Alessandra here is helping me run some of the instruments for gas concentration. So how much are they breathing? How much oxygen and carbon are they cycling? So I'm able to pair that measurement with who's there and how much cycling are they doing to get an idea in a year's worth of space and time, how that's changing. And what do I do with this? Well, the project that I'm working on right now is looking at how microbes influence net productivity, so net carbon fixation in the Arctic. And I actually use this by taking all of those measurements and applying machine learning algorithms to look for patterns within the data. And so because I have a year's worth of data, I'm actually able to use some of these more advanced statistical techniques to kind of pull those connections together. And why am I interested in that? Well, the efficiency of how much carbon gets actually sequestered into the deep ocean, so moved from the atmosphere to the ocean, is influenced by our net microbial productivity. And this is because it's controlled by the metabolic balance of that photosynthesis and respiration processes. So if this is an image of the microbial loop, so we have our phytoplankton fixing that CO2 into their biomass. Some of that gets eaten by zooplankton, some of it gets consumed by bacteria. Whatever's left gets pumped to the deep ocean for long-term storage. And so this net community production is what NCP stands for. Forgot to put that on the slide. But this net community production is essentially equal to that gross primary production, so that gross fixation, that gross photosynthesis, minus all of that respiration, so all of those loss terms. And at long-term steady states, this balance is about equal to the net carbon, which makes it to the deep ocean, neglecting eating, higher trophic levels, and everything else. And so if I'm able to measure this value and compare it with the diversity of life that's present at any given time, I'm hoping to be able to quantify some of the biological mechanisms which are changing that net value. So I'm looking at how the influence of diversity impacts that net carbon sequestration into the deep ocean. And again, I do this using machine learning models to try and predict some of the complex signals which are embedded within that data in order to extract patterns of both how the community structure of our microbes are changing and then how that influences those biogeochemical signals that we want to measure, like net community production and long-term storage. And so what my data actually looks like, this is just an example of some of my results. So this is, if you were to imagine if we took this section of drift and looked in the water column, so this is the upper 100 meters of the water, basically taking a section of the ocean and then mapping what we measured from each of those CTD casts. So from each time we collected water, we have all of our sample points, which are all of these dots. So this is all of the water that I collected from each of those casts over mostly from March through September when we had people on board. And I'm able to look at who's there. So I have each of these points colored by a functionally distinct microbial community. So lumping together more similar communities and basically giving them a name. And we map that on top of oxygen utilization. So in addition to measuring who's there, we're also measuring how much oxygen is being biologically produced or consumed. So this is a positive or negative measurement in micromoles of oxygen per kilogram of seawater. So we have, where it's positive, we have more photosynthesis occurring, and where it's negative, we have more respiration occurring. So more oxygen either produced or consumed. And what we can see is that this actually aligns really well with the presence of these functionally distinct communities. So we're starting to see communities that are associated with higher oxygen production, or communities that are associated with deeper water where we have more consumption. And what's interesting for me here is that you can see during different periods of the year, we see a lot more respiration occurring just beneath the productive surface layer. And what that tells me is that a lot of the carbon that's being fixed in the surface ocean is not actually making it to the deep ocean. It's being consumed pretty rapidly, especially during periods of the year where we have summer melt. So all of these wiggly lines, this is density. So I'm just tracking how stratified the water is. And this is, July, August, we have a lot of sea ice melt. We're pumping a lot of cold, fresh water into the surface of the Arctic Ocean as that ice melts. That floats on top of the denser, saltier water. And all of that production that's happening near the surface is getting trapped in that stratified layer. And just beneath that, the oxygen is all being consumed as that phytoplankton, which might sink out normally as it does during the winter when there's a lot of mixing, it gets consumed and that carbon doesn't get sequestered. So that's just an example of some of the results that I'm pulling together using these different methods. And looping it back to why am I interested in that? Well, if there's less or thinner ice, if there's more light, we're not really sure if that's going to equal more production. Again, we have a lot of question marks when it comes to microbes and changing climate, especially in the Arctic, where we just don't have a lot of data. But we are starting to see that there is a lot of respiration occurring. And so what we want to know is, will climate change impacts on microbial communities help or hurt the Arctic's ultimate ability to sequester the CO2 through the biological carbon pump, which I showed earlier? And all of the goal of Mosaic data, I think, taking it together as an entire expedition, so not only my little tiny piece of that puzzle, but all of the different measurements that are taken, the overall goal is to really try and understand and improve our models of this change. So this is a graph showing the projections of warming by the end of the century for all of the different greenhouse gas scenarios, which were used during the latest IPCC report. So if we have our predictions of change based on all of the different models and all of the different greenhouse scenarios, no matter what the scenario is, the uncertainties are the most big in the Arctic. So we have a really hard time actually making those predictions, mostly due to the fact that we just lack data from this area. And so the goal of Mosaic was to really get all this data in order to get a baseline to improve our models. And how my data falls into that, because I'm trying to understand these shifts in community and chemistry, I'm hoping to provide this information to better model our shifting ecological states. And that's where the machine learning comes in, where I'm using computers to then try and look for patterns that I can then better predict. And then there's a lot of people that goes into this. So these are some of the different groups. This is the Leg 4 group of Mosaic. This is the Leg 5 group of Mosaic. This is my lab funding. I'm mostly funded through NSF, but that's it for my spiel. Now I'm really happy to answer any questions.
Emelia, maybe I can feed you a few from the chat. Would that be okay?
Mm-hmm.
Okay, great. Joyanne, who's joining us from Alaska, you kind of answered the question, but maybe if there are other findings as well. Many of the pictures that you showed are when it's sunny up in the Arctic, because you were part of legs 4 and 5. What types of differences did you see in your data during the winter collection?
Yeah, so I think one of the most surprising things in the winter collections is that there's still a lot of activity going on. So I think for a long time, people just kind of assumed that the Arctic winter is mostly dead biologically. It's, everything's either in stasis or waiting for the sun to return. But we are finding that there's actually a lot more going on during those periods than previously expected. So for example, someone who's studying chlorophyll specifically found that there was high levels of photosynthesis happening, 2 days before the sun actually reached the horizon. So maybe those organisms are really adapted to low light levels and they're really just kind of primed and ready to go even before the sun returns to actually start growing. We also found some fish that were unexpected. And in my data specifically, I found that a lot of the oxygen, which was produced during the previous bloom, so during the fall before the sun went under the horizon, actually stayed trapped underneath the ice. So we had high levels of oxygen signal far into the winter, much longer than we'd expect, mostly just due to the fact that it's kind of capped by that ice level. And so we're able to have, productive conditions far into the winter and still have stuff for things to eat. So that's just some of the few things that we encountered.
Cool, that's really interesting. Yeah. What were some of the coldest temperatures you experienced? And did anyone ever fall into the water?
Yes, there were quite a few. I don't know if I have a picture of it. I don't have a picture of it in this presentation, but ice is pretty treacherous. And so there are a lot of plans for when people were to fall in the ice, what was going to happen, etc. When I was there, it was very warm. So I think the coldest I experienced was probably like -15. So not that cold comparatively. But during the winter, it got very, very cold. I think it was like -40, -60, crazy cold temperatures. So the poor winter folks really had it a lot tougher. But there were some instances of people falling through the ice. And I think my favorite is, my team leader on the expedition, Ali Fong, at one point, she fell through the ice. And then for the rest of the expedition, she fell right near an ice ridge where 2 plates of ice kind of bunched up. And that became known as Ali's Ridge for the rest of the expedition. So if you ever see that in a publication from Mosaic, know it's named that because Ali fell through the ice.
Infamous. That's great. Janine has a question about how you got back and forth for the expedition for your legs of the 5.
So there was we essentially had support icebreakers was the plan. And so we had other ships that would come and sail up kind of close to where Polar Stern was locked in, and then people would transport via snowmobile across the ice, all the people and the stuff and the food and everything. But COVID threw a huge wrench into the entire expedition. I mean, they were up there when it started because they were started in 2019, and I went up in April of 2020. Where we weren't really sure how we were going to do that transition because a lot of the different countries pulled their icebreakers out. So we ended up having to go with other German ships. They were the only country that was still like, we're making this happen no matter what. but we weren't able to have icebreakers. So there was a period of time, and you can actually see it in my data, where there's like a gap in measurements, and that's because the polar stern had to leave the ice camp, meet up with another ship where there was open water right near Svalbard. So when I did my transition, I flew to Bremerhaven, had a quarantine there. Then we took a ship from Bremerhaven to near Svalbard, but we weren't allowed on shore. And then we did a transition and got refueled there. So then we went back up north and stayed with the flow for the rest of the time and found it using GPS coordinates of instruments. So there was a couple of days where we were like, all right, I hope we find our stuff. But we did, and ended up being fine. And we got really good about putting down and taking up ice pans.
That's crazy, Emelia. I imagine, I think it was what, 2 weeks of quarantine in Bremerhaven before you were.
Yeah, I had to, and it was before COVID tests were really accessible either. So I had to have a COVID test shipped to me in San Diego from Germany, and then find a doctor to administer it all during lockdown. So,
Yeah, what a crazy time that was, for sure. Emelia, another question is, are you going back to the Arctic anytime soon?
So I was actually just up in Alaska, not on land, but on shore. I did a transit from Seward to Nome in June. So that was really fun. And I got to collect some more data from the Pacific side of the Arctic. I don't have any plans after that, but as I said, I'm wrapping up my PhD now and transitioning. So I do plan to get back up to the Arctic to continue this research. And actually, I've now constructed a couple of models, which I want to collect some more data to validate. So I'm hoping to, but I don't have any plans right now.
Great, thanks. Sandy, do you want to hop on and ask your question?
Yeah, thanks. Hi, thank you so much for your presentation. I really am just curious of kind of the organization of this expedition in terms of, you said you were there during midnight summer, like the summer period of time. Was that on purpose, or were there certain scientists that were brought in during the summer versus the winter? And also I was wondering, since everyone, all the scientists have come back from this expedition, are you still sharing a lot of your data? Are there future plans to maybe get everybody together again? Is there like an overarching goal of what everybody did on this? You talk about there's a lot of different countries that were involved, a lot of different scientists involved, studying really specific things that were related to the Arctic. Is there an organization that's kind of putting all of that together?
Yeah, that's a great question. And this is, I think, one of the thing that was the most impressive to me for the expedition leaders, Matt Shoup at University of Colorado Boulder and Marcus Rex at the Avi, and they still continue to organize a lot of those types of activities. So we've actually had multiple science conferences since then, where everybody that can, so not only people that were on board, but also all of the people who are analyzing the data and presenting it and different components of that. We've now met up twice. So there was a science conference in Germany in 2021, and then there was one in 2022, and then there was one in Boulder in 2023 in this past February. So we have continued to meet up as a large group and share our data, present it, try and build collaborations between what was connected to build more interdisciplinary studies. All of the data is online at this point. So that was part of if you were involved in Mosaic, you had to make your data publicly accessible. So a lot of it is up on different databases with a Mosaic tag so that people know it's all related. And then even beyond that, I mean, I still have team meetings every month to 2 months. We meet virtually online within our different groups. So I attend both the ecosystem and biogeochemistry team meetings on Zoom. where we present new results, talk about continuing analyses. We're a little behind in analyzing our data in the ecosystem team just because it takes a little bit longer than I think some of the physical measurements. But we're still very much actively involved in doing this as a collaborative research endeavor, and trying to get out not only just our own tiny pieces, but more overview publications on either specific topics or teams, which are available as well. So it's impressive that they planned with this longevity in mind.
And did scientists have a choice as to what period of time or what leg they decided to join the expedition?
So I think it had a lot to do with what your project was funded to do. So all of the scientists on board had their own funding from their own countries to then, leverage this platform. So for example, I mean, we had personnel on board for legs 3, 4, and 5, and we had a colleague who was going on leg 1 during the initialization, and she was kind enough to actually collect data for us as well. So we have some data from the fall and winter. So it, even if you couldn't get a personnel spot, just because spots are limited, we had a very team mindset where we'd try to collect data for everyone during all legs. So there was like a couple parameters which were, these are really important. We need to get it from every single leg if there's a person on board or not. And then there was some where it was a very specialized project. So example, there were some people who were really studying phytoplankton. So they only came on board for legs, basically the summer legs, because during the winter, there wasn't as much that they were interested in. So it really depended on your research question and then the logistics involved.
Great, thank you.
Thanks, Sandy. Emelia, Lolly was wondering, let me find the chat. I think this is a fun question. If there were any issues with ridging for the ship or for instruments, those ice ridges you were referencing.
Yeah, so it's a constant struggle. There were more than a few instruments that were unfortunately lost due to ice activity. Especially, there were less so when I was on board because the summer was a little bit calmer. It tends to be more stable meteorologically. But during leg 3, which was from February to April, so very strong winter period into spring, there was a lot of ice activity, and at one point we actually lost half of our observatory. So when I came on leg 4, the central ice observatory was much smaller than it had been when my advisor was on board. So it was pretty funny because they kept being like, oh, we'll go to this site, and we'd be like, that site doesn't exist anymore. It was eaten. And so there was a lot of, you're constantly monitoring the ice, both when you're on it and when you're on board the ship, in case something like that does happen. And we had, in addition to scientists, I mean, there was the ship's crew as well as some dedicated logistics personnel who were in charge of making sure all of the electronics were working, the cords were set up, there was data transferring between the instruments and the ship. And so whenever something like that would happen, they'd all rush out and stabilize and try to recover what they could of the ridging and make it more feasible. But it is an extreme environment, and there is always that inherent risk of ice doing what ice does. But it also provides for some really interesting measurements, especially for the chemistry side of things. If you have a stable ice sheet and then all of a sudden there's a big hole, or there's a huge ridge. And there's actually a project that was, their whole goal was to study ridges. That was their entire science plan, was finding and sampling ridges. So yeah, a lot going on, especially during the winter.
Yeah, pretty crazy to have such a dynamic field site. I'll have to share with the group. I'll share a link to some photo libraries and some of the ridges, and the instrument consumption by those ridges are pretty amazing to see, for sure. Emelia, do you have, you share with us maybe your biggest personal experience, biggest takeaway personally from the Mosaic experience?
Yeah. Well, I guess two things. So the first was actually just how dynamic the ice was. Even on the summer legs, when I was there, we had a lot of changes going on and occurring around us due to melt. And that was really incredible to see in person. I mean, up until this was my first time, this was my first time in the Arctic proper, as well as my first time on a ship. This was actually my first ship-based expedition. So I went from 0 to 100 with 6 months. But it was really interesting to see what I read about in papers in person and the dynamics that were happening, because we'd go out one day and sample a melt pond, and then the next day we'd go out and it would be gone, or it would be 10 times bigger. And for me, I'm interested in a lot of those processes on the micro scale. So thinking about how sea ice gets colonized by microbes, where are they living, where are they most productive, and if the environment is changing that rapidly, there's a lot more, I guess, heterogeneity to consider than I had previously been thinking about in my science. So from the personal experience of just being there and actually seeing it in person, I think that changed not only how I look at the Arctic, but also how I look at my research. So that was pretty incredible. And then again, it was my first big research expedition. I'd been to the Antarctic, so I'd done some camping in the Antarctic dry valleys, and the differences between that expedition and the ship-based work was night and day. And I just really love the ocean, and just being out there for a period of time and being able to see what is truly a beautiful environment kind of made me, fall in love with it all over again. And I definitely want to keep doing this for forever. So I guess that's a little bit life-changing in a way.
Thanks for sharing, Emelia. Yeah, can only imagine what it must be like to be doing work in those remote regions. I bet it's pretty special. Joanne wants to know, she knows that scientists are pretty crazy, all caps, LOL, JK. But she wants to know, besides eating chocolate, what did you all do for fun up there?
Yeah, so it seems like scientists pretty well. So, I mean, there were definitely some football games that happened on the European football, because there was a lot of Europeans on board. So a lot of soccer games that occurred on the ice, I think, during downtimes. We tried to schedule the days such that it was sustainable, because it was going to be such a long period of time. So we only worked on the ice from 9 a.m. to 5 p.m. and all the rest of the time had to be spent on the ship unless you got special permission to study something. And on Sunday mornings, there was always no ice work. So it was always a period of intentional downtime, because we were out there for months at a time. So they're really trying to make sure we stayed safe and not exhausted, because, give scientists the chance, they will just keep working the entire time. And the logistics team was like, no, you have to actually rest and eat ice cream and watch a movie and be a human in addition to your work. So we did have some, sporting events. There's a gym on board. We'd have movie nights. We'd have barbecues to celebrate holidays or birthdays. So, you're on board for such a long period of time, you end up having a lot of birthdays. So there was a little barbecue that we'd bring out on deck. So midnight sun, it was beautiful all the time. You'd stand up and barbecue up your German sausages or potatoes or whatever. And we'd have music. There were people on board who brought instruments and would actually play. So sometimes we'd have live music. So it was a nice time as well. I was in the lab for most of it, but it was a really just overall, I think, exciting experience, not only because of what we were studying, but the people we were on board with.
Cool. Yeah, thanks, Emelia.